Hello, everyone. I'm Dr. Marie McNeely, and I am here to welcome you to episode 609 of the People Behind the Science podcast. Listeners, thank you for tuning in today. If you haven't had a chance to meet our guest, Dr. Jan Westpheling, already, we are rebroadcasting our interview originally published on August 7th of 2015. Listeners, Jan is conducting fascinating research in the area of biofuels. Specifically, her lab is working to engineer bacteria to convert plants into fuels and other useful chemicals in order to address our energy needs in a more sustainable and environmentally friendly way. And in our interview, Jan gives more details about her science, her career, and her life outside the lab. So I hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. Today, I am excited to be joined by Dr. Jan Westpheling. So, Jan, thank you so much for joining us today. How are you?
I'm fine. It's a pleasure to be here.
Well, it is a pleasure to have you with us today. Listeners, just a little bit about Jan before we hear her story. She is a professor of genetics in the Franklin College of Arts and Sciences from the University of Georgia. She received her bachelor's degree in microbiology from Purdue University. And Jan worked as a research technician at Eli Lilly before getting her Ph.D. in genetics from the John Innes Institute. She then worked for a year for Biogen in Geneva and conducted postdoctoral research at Harvard University before joining the faculty at the University of Pennsylvania Medical School, where she served for a brief time before she came to her current position here now at the University of Georgia. And now Jan is the two-time recipient of the Creative Research Medal from the University of Georgia, and she's also served as a member of the National Research Council Committee on the Development and Acquisition of Medical Countermeasures Against Biological Warfare Agents in 2005. And Jan, you are here with us today to share your journey through life and science. So myself and our listeners want to get to know you, both as a scientist, but also just as a person. So can you start off by giving us a window into your life outside of science and what you like to do?
In the 5 or 10 minutes a week I have outside my office.
Sure.
I do take some time. I love gardening. I'm almost to the master gardener status, but not quite. I live on 8 acres of old growth forest, which is only 10 minutes from my lab. And I have made my woods a kind of bird sanctuary. I'm one of these crazy people who plants trees in the forest. But I plant trees that birds like to eat berries from and things like that. So I have bird feeders everywhere. I have owls and all kinds of migratory birds that come through every year. I really enjoy being in the woods and enjoying the wildlife. We have deer, raccoons. A fawn was born in our yard last month. It's just really nice to be able to live in an environment like that, so close to work.
Well, that sounds absolutely picturesque, Jan. And I'm curious, you mentioned you're almost to the point of a master gardener. What is the one thing that will bump you over that threshold?
I have to learn genus and species names of plants. And in my yard, I think there are 20 different varieties of oak trees.
I didn't even know there were that many, so that's impressive.
So I'm not quite there yet.
Gotcha. Well, fantastic to hear about what occupies your time when you're not at science. It sounds like you keep quite busy in the garden, enjoying your yard. And I think it's wonderful to kind of get outside, get away from the lab, get away from the science for a little bit. But we're only going to let you get away for a little bit. So we want to talk about the science next. So can you tell us how you describe what you do at work to someone who's not in science?
Well, for me, that's easy because I work on biofuels, and you can hardly pick up a newspaper these days without reading about climate change or the need for a carbon neutral, sustainable energy source. It's in the popular press, it's on TV, it's in social media. So people are constantly hearing about biofuels and bioenergy. So when I tell people that I'm interested in using non-food crop plants, not corn, crops like switchgrass as feedstocks for the conversion of those plant biomass materials to ethanol and other fuels derived from plant sources, and I engineer bacteria that can do this, they understand that. I think my non-scientific family finally thinks I'm doing something useful.
And something very cool, I must say. So then what is the advantage of switchgrass versus corn, or why would you want to use switchgrass instead of corn?
Well, corn is a food crop, obviously, and you don't want to engineer a food crop. I'm part of one of the bioenergy science centers. It's one of 3 major centers in the United States, and our mission is to engineer plants to be less recalcitrant to digestion by microbes, and we engineer microbes that are engineered to use plant biomass and make it into compounds of interest like ethanol or butanol or isobutanol. And so switchgrass will grow in the medians of highways. It's a perennial grass. You mow it, harvest it. It's one of the bioenergy crops that have been designated by DOE that we can engineer. We can weaken its ability to resist deconstruction by microbes, and that's fine. You can't do that with a food crop. And using a food crop for fuel obviously is already having an economic impact on the costs of food in places like the third world. It's just not a desirable thing to do for lots of reasons. So switchgrass and populus, sorghum, miscanthus, those kinds of crops that are not used for food are good sources of biomass.
Definitely. Well, great to hear a little bit about your work, Jan. And we'll talk throughout the interview about some exciting projects that you've been working on. But I want to take a pit stop here and talk about motivation. I think sometimes it's hard in the day to stay motivated when you have so much on your plate, so much you would like to get done, things that you're excited to do. Do you have a particular favorite quote or a saying that you keep in your mind to get you through it all?
I love my job so much that I literally pinch myself in the morning and say, they're going to pay me to do this.
I like that. You're lucky.
I am lucky. I heard a quote when I was a graduate student that I've always found really funny. It's, all that glitters may not be gold, but at least it has free electrons. And I've always thought that was fun. But to me, it means that it doesn't have to be gold to be important and to be worthwhile. You know, not all of us who do science are going to win Nobel Prizes, nor should we win Nobel Prizes. But everyone has something to contribute. And even the smallest contribution is important. And so we should pay attention not just to things that are published in Science and Nature, but smaller bits and pieces of work that contribute to a bigger whole. And I think that's important, not to dismiss things because they're not gee wow. It's also important to note that people hit their strides at different times, and not everybody does it in the same time frame. Because I worked for a couple of years in industry after college, and I was at Biogen for a year, I always felt like I was a little bit behind my cohort. I have to tell you a story about one of my favorite people, Carol Gross. She's a dear friend and a great scientist, and she came to give a seminar at UGA when I was an assistant professor, and I'd known Carol since I was a graduate student. She's one of those people that you just are drawn to in science because she's so real. And she actually stayed at my house, and we're sitting on my back porch having a glass of wine, and she said, "So, how are things going, Jan?" And I said, "Well, you know, Carol, I feel a little behind." And she said, "I got my first job when I was 40. They offered it to 5 people before me. And she was a member of the National Academy of Sciences when she was 50.
Oh, wow.
So I thought, I got time.
Right. You can make this work. Yeah.
But she convinced me that it's not how fast you get somewhere, but that you end up in the right place.
Well, I think that is such an important lesson, and important for people who might be following a quote unquote non-traditional career path in science to get to where they want to go. They feel like they are behind. You have that thing where you're kind of looking at everybody who's starting their families, who's feeling like they're way farther on in life than you are at that moment. But you are exactly where you need to be. You just have to make it work with what you have and keep going forward, I think is so important.
Exactly. And if you're not enjoying the journey, you're probably not on the right path.
Definitely. Well, I love this piece that you brought up about what you're working on might not be the hottest thing right now in science, but it might be important down the line, I think is something to think about as well. You never know where the different directions of other fields are going to go, so that your piece might end up in the spotlight whether you realize it or not.
Well, the entire biotechnology industry is based on the discoveries of people like Hamilton Smith, who was working on restriction enzymes because they imparted phage resistance to E. coli. No one ever imagined that restriction enzymes would be the key to cloning and genetic engineering. So I think there's something to be said for doing what you love and trusting that it will make a contribution.
Absolutely. So now I'm curious to hear about who has inspired you along your journey, Jan. Can you tell us about some of these big scientists who've really made a big impact on you? And this can be historical role models or even people who you've looked up to along your journey.
Well, this is going to sound a little strange, but I don't believe in role models.
Interesting.
I think there's a big difference between role models and mentors and teachers. And I've been the beneficiary of wonderful teachers, and I've learned a great deal from them. But we're all different people. we can watch people do things and we can understand the decisions they've made, but not ever really understand what it's like to be in their shoes. I've learned a lot by watching people do the things that I want to accomplish. But I learned from people like Carol Gross and Lois Miller that you have to do things in your own way and on your own terms. And so I think we can't abdicate the responsibility to find our own paths by looking too closely at what other people do and how they did it. We have to choose our own paths and figure it out for ourselves and not get too wedded to the idea that whatever somebody else does will work for you.
Certainly. And I think a lot of the challenge is finding someone who will support you in doing what you want to do, which is, you're right, what a lot of these teachers and mentors are going to do for you. So very cool to hear your take on role models. And I think you're right. A lot of scientists don't have this exact picture, this idea of the person that they want to be or the things that they want to achieve along their career. So it is important to kind of strike it out on your own and do what inspires you. So now can you take us back to the beginning, Jan, and tell us what really got you started on the path to science? Do you have early memories of maybe as a child being interested in science, or is this something that developed later on?
I've wanted to be a scientist since before I knew what that meant. I grew up in a small farming community in the South. I did not come from an academic family. My mother's Italian parents were not literate in English. I went to a grade school that didn't own a microscope. But I was always fascinated. When people would say, how does the grass get bigger? I wanted to understand that at the molecular level. And I'm dating myself here, but I used to watch a program called Mr. Wizard. It was a weekly program. And when you lived in the middle of nowhere and there were no like science museums or anything like that, this was a program that came on every week. And he did experiments with kids. And he had a laboratory. And he was always surrounded by little kids about my age. And he would do experiments with them. And it was absolutely fascinating. Little things like converting steam into liquid water by condensing it, a pie pan over a boiling pot of water. And then he would explain the chemistry of that. And I was absolutely fascinated by that. And so as I was growing up, I was a science nerd. I went to Purdue, where my genetics professor was a man named Ed Simon, and he was a really famous virologist, and he was my genetics professor. And he had a novel approach to teaching. He made up these organisms. They were from Al Capp's Schmooze. Do you remember this?
No.
This is all before your time.
I'm going to have to look this up later.
Al Capp was a cartoonist, and he had these creatures in his cartoons called Schmooze. They looked a bit like Frosty the Snowman.
Okay.
So Ed Simon invented this genetics course, which is internationally famous, and he would give Schmooze whatever genetic characteristics he wanted to. For example, he would make up chromosome structure. He would make up the number of chromosomes, and he would make up dominance and recessive relationships, and he would make these things up. And then he would test you on your ability to think through a problem. And there was no memorization involved because it wasn't real, but it taught you to think like a geneticist. And I loved it. I mean, I think in every person who's been somewhat successful in their career, you can point to a teacher and say, that person really taught me to think about this the right way. After college, as you pointed out, I was a research technician at Eli Lilly, and I had wonderful mentors there. I was young and brash and outspoken and not always happy with a conservative approach to dealing with things. I had wonderful mentors there who put up with me, Larry Day, Dick Bolt, Steve Queener. These were guys who taught me to use a little tact to temper my enthusiasm.
The softer skills, yes.
A softer, gentler version of who I was before. So being in that kind of industrial, professional environment was really good for me when I was growing up, because it taught me that being effective was just as important as being right. If you're not effective, it doesn't matter if you're right. So I learned some good lessons there. I was really fortunate to do my Ph.D. in England with David Hopwood, who is a fabulous bacterial geneticist. He ran a very small lab. He provided daily attention to his graduate students. He knew every experiment I was doing. In his mind, he had a little clock, and he would say, okay, well, if she works really hard, she'll know this on Tuesday, and on Wednesday, I'm going to ask her how it went. And the challenge was to tell him on Monday. I had so much respect for him. He taught me a great deal about how to train students, that respect is something that you earned. You didn't just have it because you were the professor. He also taught me that true leadership depends on leading by example. And no matter what you say to people, it's what you do that counts. I once asked him how I could possibly repay him for all he'd done for me, and he said, that's not how it works. You don't pay back to the people who've helped you. You pass on to your own students whatever they've given you. And that's our real legacy in science, is the students who come after us. They're smarter. They're going to go on well after we're retired. And the real contribution that we make in science is not so much what we do in our own labs, but the people we produce that we're going to go on and do even greater things. After graduate school, I worked for a year in Geneva with Julian Davis. And I think that experience convinced me that I wanted to be an academic. I did not want to work in a company. And, I think getting these kinds of experiences with industry can teach you as much about what you don't want to do with your life as about what you do want to do with your life. I don't deal well with authority, so working in a company was just not enough.
You have to be your own boss.
Yeah. Be my own boss. I then went to Rich Losick's lab at Harvard. So I'd been a graduate student in a lab of 6 people, and when I joined Rich's lab, there were 15 postdocs and about a dozen graduate students.
Wow, big change.
Big change. Rich was tough but fair, and taught me a lot of what I needed to get along in a competitive environment. And science can be a tough business. We had joint lab meetings with Jan Perro's group at Harvard. Wally Gilbert was around the corner. It was a fantastic environment, the students and postdocs there. We had joint lab meetings with Jan's group, and Rich was a bit of a cheerleader, and we'd always look over to Jan, and when she smiled, you knew that you'd nailed it. And they've continued to be good friends and good colleagues and helped me all along the stages of my career. And, of course, I've benefited from interaction with amazing colleagues and people who've taught me things I don't know. One of the things about being an academic is you don't ever have to stop being a student. I work now in this Bioenergy Science Center with people who do such different things. I mean, until I interact with these people, I wouldn't have known an NMR if I were sitting on it.
Right.
And I work with plant cell wall chemists, and it's just been a fantastic journey as a student.
Definitely. Well, Jan, I think you brought up so many important points here, and I'm going to take us back to one of the first ones you made, just this idea of learning how to think like a scientist and how a teacher's particular approach to learning, making up these imaginary creatures with imaginary genetics and rules you had to follow. I think that is a really important approach in science that is really underutilized. I mean, I think the focus is so much, particularly at the undergraduate level, on memorization, on learning the basics, learning the foundations, which I think is important. But then if you never learn to take that next step, it's really hard in graduate school when suddenly you're expected to.
Yes, I teach large classes of undergraduates. I teach genetics, and we flip our classroom for about half of the semester. And I don't know if you're familiar with flipping, but it means you listen to the lecture and you read the textbook outside class. We record the lectures, and then in class you do problem solving. And I was a bit skeptical about this entire approach, but I think it's really good. I don't think it's going to substitute. If you want to be a concert pianist, you've got to learn the notes, and that's important. But I think learning to use the notes and make music requires really thinking and getting away from a prescription-based teaching method. I really love teaching. It's one of my passions in my job, and the students are always smart. You never really understand something until you teach it. Mm-hmm. The first year I taught genetics, I learned all kinds of things I thought I already knew, but I really didn't. Because it gets down to the nitty gritty, and some undergraduate raises their hand and say, yeah, but how does that really work? And you think, yeah,
I don't remember reading that. Yeah. Well, Jan, great to hear about how you've enjoyed teaching over the years. And I think it is such an important part of the job of a professor. And I'm interested to hear about your experiences. You mentioned kind of after that one experience at Biogen, you knew the industry wasn't for you, but a lot of people are still trying to make this decision. And it's tough because in a lot of cases, they've seen what an academic position looks like, but they haven't really seen what an industry position looks like. So were there some main things that you can point to that really differentiated the two?
Stress is part of the job in any profession, whether you're a lawyer, a doctor, a scientist, business person, whatever you decide to do with your life, if you want to do it well, it's going to bring stress into your life. It's the kind of stress you feel most comfortable dealing with. And yeah, in academics, there are a lot of stresses. It's recruiting money for your lab. You walk into a job, someone hands you a textbook and said, now you're a teacher. Mm-hmm. You recruit graduate students and now you're a personnel manager. And you've got to recruit grants. You've got to keep the science going. You've got to do all these kinds. And there's a lot of uncertainty in that. There's no question about that. But it's also very exciting. Writing a grant means that every 5 years, or even more often than that, you got to really rethink everything you're doing. And it's got to make sense. And that's a good exercise. Industry has different stresses, and I can only speak for myself, but I didn't like being in a hierarchical structure. I didn't like the fact that I could walk into work one day and they say, you don't work on that anymore because the company's interests have changed. You're reporting to this person who doesn't have a clue about what you do. When I worked at Lilly, I had fantastic bosses, but a lot of them were chemical engineers, and they really didn't understand biology. They were great people, and they understood what they didn't know, but that's not always true. And so the frustrations of working in a company for me would have been just much more difficult to deal with.
Gotcha. Well, thank you for sharing your insights there. And I think that is helpful for people out there who, like I said, might be in this decision point where they're trying to figure out if it's right for them. And I think in a lot of cases, you're right. You sometimes just have to try it to really know whether it's a good fit for you.
Absolutely. We work on industrially related topics in my lab, and I encourage my students to do internships at companies to test it out and see if they like it. Sometimes they come back and they say, this is for me. Sometimes they come back and they say, no, this isn't for me. And the experience is what you really need to do. So if you're a student, I tell my students this, if you're doubtful about what you might want to do, go test the waters out there and try out some things. Again, it's not how fast you get to where you're going. It's just important that you get to the right place.
Definitely. And I think there's some concern out there that there might be a stigma in academia against hiring someone who maybe has worked in industry for a number of years. Did you experience any of that when you were going through then trying to get back into the academic world for a postdoc?
No, but there's difficulty. Let's say, for example, I had taken a job at Biogen and I'd worked there for 5 or 6 years after my postdoc. And then I wanted to get back. As an academic, I can tell you that when you look at people, sometimes it's difficult to identify their intellectual contribution when they're working in an enormous group of people. Success in academics, you really have to live by your wits, and you really do have to run your own cottage industry. So I think the kinds of concerns that you hear around the table when we're considering hiring people from an industrial background is like, can we distinguish what they did from what, say, the army of chemists down the hall contributed to their project?
Sure.
So it's certainly not impossible. And I think those lines are being blurred more and more all the time to the benefit of both industry and academics. But it's harder. And I tell my students this, once you've gone to industry, it's harder to come back. It's always easier to get there from where you are in academics, but it's a little harder to come back. At least that's been my experience.
Gotcha. Well, thank you for sharing those insights. I think this is a question that is, like I said, on a lot of young scientists' mind who might be trying to chart their path and figure out what steps make the most sense for them to take. So thank you for that. Now, we've talked about your journey to get to where you are today, but I want to talk now about your current position and some of the work that you're doing. So, Jan, can you share with us the project that you have on the table right now that you are just so psyched about?
Well, we're geneticists, and the projects that we're working on now are part of this bioenergy science center I described to you. And our mission, the center's mission, is to engineer plants to be more easily deconstructed and to engineer bacteria to use those plants to make fuels and products of interest in a sustainable way. And so we work on an organism called Caldicellulosauropter beskyei. And
what a name there, yeah.
I know. So Caldi has a very important characteristic. It already knows how to use biomass. So if you literally take switchgrass and grind it up, add a mineral salt solution, it'll grow on it. And that's the hard part. Eating biomass is the hard part. So what we've been doing in my lab is taking organisms like Caldi and developing methods for genetic analysis. So Caldi can do this great thing, and that's the hard part. But in order to make it useful, you have to be able to manipulate it. So for this particular organism, we developed genetic methods that work for it, and we ran into some real roadblocks. For example, restriction of DNA from E. coli was an absolute barrier to DNA transformation. So we identified the restriction enzymes that were important. We knocked them out. We developed methods for our transformation of the strain. We invested some time in developing these genetic systems. And then once we'd done that, teaching it to make ethanol from switchgrass was relatively trivial. We published that in PNAS about a year ago, and it was the first example of using a non-food lignocellulosic biomass substrate, converted, unpretreated, no enzymes, no pretreatment, which is an economic barrier to using biomass because those pretreatment technologies are very expensive.
Sure.
But being able to directly convert it to ethanol by knocking out one gene and creating a synthetic pathway by cloning in another gene, we were able to make ethanol at 60% yield.
Wow.
I'm very excited about that. So we are continuing that work with these organisms. And we're also, within BESC, initiating new projects to take what we call uncharacterized organisms, as Lee Lin calls it, organisms out of the mud. So taking these organisms that can do things that are impossible to engineer, like grow at pH 2, or do some way cool chemistry that allows them to use substrates that you can't engineer in these pathways because of redox or an inability to metabolically support the pathway you want to engineer in. Taking these organisms and developing methods of genetics that will instantly open them up to manipulation. So we're working on developing genetic tools that will be generally applicable to these organisms. So quality, for example, is an obligate anaerobe that grows at 80 degrees centigrade. And those kinds of issues make it a little more difficult to work with. But the main thing is to get genetics working for a lot of organisms that do the cool stuff.
This sounds fascinating. So thinking about the process, the big picture, how far are you away from, let's take this project on collie, for example, getting that work out into some sort of translational component?
We've extended the genetic tools that we developed for them to the Clostridia. We're working on Thermoanorobacter. So these tools, we just recently discovered that all of the plasmid vectors that we developed based on native quality replicons function in all these thermophiles that we never expected. So this promiscuous plasmid replication ability in a large number of thermophiles is going to open up genetics immediately. It gives you a tool for transformation. It gives you a way to express heterologous genes, open up pathways, knock genes out. So that's turning out to be surprising that we can extend these genetic methods to other organisms. And of course, we're always looking for new kinds of replicons, new methods. Restriction is a huge issue in these organisms. There are restriction enzymes that are unique to these genus. And that's also a little surprising. So it's fun.
Definitely. So when can we expect, would you say, some of these enhancements in biofuel creation and use?
We're already moving forward in that direction. The paper we published in PNAS last year sets the stage for the direct conversion of switchgrass to fuels like ethanol and butanol. The yields and titers need to be improved, but it's the basis for engineers will now come in and take over and do the difficult work of making it an industrial process. But that's already in progress.
Gotcha. Well, so cool to hear about the progress that you're making on this line, Jan, and wonderful to hear more about what is going on in your laboratory. And we've hinted through this discussion of some of the roadblocks, some of the challenges that you encountered in these genetic systems. But can you think back now on your career and tell us about, maybe in detail, one of these specific examples, or just one of the challenges you have faced, one of the failures you've had, one of the tough times in science? Because I think the public doesn't always realize the path of a scientist is littered with these challenges, these frustrations, these failures, but you have to push through them to continue being successful. So can you share a story with us?
When you use words like struggle and major failure, that's a personal question, and I'd like to answer it with a personal answer.
Right. These are my nightmares as well. So
I personally struggle all the time with feelings of intellectual inadequacy, and I'm going to wake up one morning and they're going to realize I'm a fraud. I can't really do this job. I have doubts about my ability to do science.
Right.
I was at a faculty meeting not too long ago, and we had these job candidates come through, and I said, the only honorable thing I can do is resign so you can hire two of these people. My colleagues around the table go, good idea. But seriously, maintaining a sense of confidence in the face of things like your grant gets triaged. You've spent 3 months putting everything you hope to do down on a piece of paper, and your colleagues don't find it worthy of review. That's tough. Feels like you've been kicked in the gut with a steel-toed boot. But it happens. It happens to everyone, and you just deal with it and move on. I think the most difficult thing I've had to deal with in science, I'll call them science bullies. Mostly I've had a great experience with my colleagues. They've been supportive and helpful, but every now and then you run across someone who takes advantage of situations to intimidate people because their science isn't enough to silence their critics. And so they do things that are not what you would like them to be. I'll just say it. They do things that are somewhat unprofessional. And at those times, it's been really hard for me to fight back. I'm not very well equipped to deal with things like that. But I've learned that the best way to fight back is to keep your head down, get on with your science, and let it speak for you. Lois Miller taught me that. She was a dear friend and colleague in my department. I'm a fanatical football fan, all kinds. And on my wall in my office, there's a frame-by-frame account of the catch. if you're not a football crazy person, you probably don't know this, but Joe Montana was a quarterback for the San Francisco 49ers. And during the playoffs, they played the Dallas Cowboys, and Too Tall Jones just trash-talked the entire week before the NFL championship game. And he said he had no respect for Joe Montana. He was a fifth-rate quarterback. The Cowboys in the season had been a fluke, that the guy was a joke, yada, 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 yada. And at the end of the game, Montana threw the pass over Too Tall Jones's head to hit Dwight Clark in the end zone and score the winning touchdown. And he got knocked down in the process. And Joe Montana's laying on the ground, and he looked up at Too Tall Jones and he said, "Respect that."
That is a great story. I wasn't a football fan earlier, but I might be now.
So when people put you down, you just keep your head down and get on with what you do and let what you do speak for you. That would be my advice.
Well, Jan, thank you for sharing these experiences. I think these are really important things to prepare people for who might be going into a career in science. There are these people who just do not play well with others, and you can't let them keep you down.
Yes. And you can't let it hurt you. You can't let it define who you are. You get to define who you are.
Absolutely. Well, thank you for sharing stories about the tough times. But next, I want to talk about the happy times. So can you share with us today one of your success stories that's meant a lot to you? And this can be a big success or even just a little win along the way.
I think our recent success with this switchgrass work has just meant the world to my students. I run a very small lab, and we are a team. And they've been so excited about this and so happy about it. I would say that my personal successes in science have been when my students have really achieved something great, like gotten the job of their dreams or published their first author manuscript. And you see how just happy they are. Those have been the most special moments for me.
Absolutely. I think knowing that you've contributed to that success is really rewarding and a meaningful part of the job that I think a lot of professors look forward to, being able to work with students and help them along their careers. So wonderful to hear about your successes, Jan. And we talked a little bit about your life outside of science. And I want to know what you read in your spare time. Jan, do you have a book recommendation for myself and our listeners today?
I'm a news junkie. And I'm also fascinated by books about the Supreme Court. So I would recommend a book called The Brethren. It was written some years ago, but it's a book about the Supreme Court. And Jeffrey Toobin now writes personal accounts of the behind-the-scenes people in the Supreme Court. I am fascinated by the Supreme Court and its impact on laws in this country, but surprisingly, the attitudes of people in this country. How quickly things can change when the law of the land becomes the law of the land. Just recently, these Supreme Court decisions have revolutionized the way the country thinks and feels about really controversial topics. So these books about the Supreme Court and the people who are the justices, I've actually interacted with a couple of them through student interactions, and they're amazing people. And learning about how their own experiences shape the way they think about the law and the Constitution, it just fascinates me.
Definitely. Well, great recommendation. Interesting that you are in this area completely outside and separate from science, but also somewhat tangentially related when you think about the science policy implications and the law and all those sorts of things as well. So great recommendation for my listeners. And we've talked a little bit about some of the different places that you've been, these different positions that you've held throughout your career in science. And in addition to training different places, scientists also just get a lot of travel opportunities to go places for conferences, to work with different people, collaborate, things like that. So can you now, Jan, think back on all of the places in the world that you have been for science and share with us one of your favorite places and why it was so special to you?
My favorite city in the world is Rome, no question. It's just one of the most friendly, unpretentious, welcoming places I've ever been. And I was a graduate student in England. As we say, we're two countries divided by common language. Learning to speak British English was a hoot.
Really?
Oh, my God. I realized for the first time that not everybody thinks the same things are funny. I would go to plays in London, and everyone around me would be laughing, and I think, I don't get it. What are these people laughing at? Mm-hmm. So I would watch evening soap operas on the BBC, things like, you won't know this, Fawlty Towers and Coronation Street. These were like the equivalent of As the World Turns.
Okay.
I learned slang. I learned what people think are funny and learning how different people view the world. We're not all the same. And that was really interesting. I learned French when I lived in Geneva, and I learned the idioms in my own language. You don't think of the idioms in your own language until you're speaking a different language.
Right, trying to translate them. Yeah.
I ran a lab in Singapore for several years, in Biopolis. I've been to China many times, to Greece. When I was a graduate student in England, you could get a URL pass for $50. You could go to Istanbul from London. I've literally been to every place I've ever wanted to go. And I've really, really enjoyed that. And what I've learned from the people I've interacted with has really been invaluable. I still do way too much traveling, but I do less than I used to before. But it's always a lot of fun.
Definitely. And I love the story about almost the language barrier, even when you're speaking the same language. I think a lot of people have encountered this. When you go to different countries, or if you're maybe an undergraduate and they're international students, there are just even words that they say that you're like, that's not what we call that. But
they use the same word to mean something completely different, and you think you're speaking English.
Right.
I bought a car, and suddenly there was the silencer and the bonnet and the wing. And I'm like, what are you talking about? Mm-hmm. So it's the fender, the muffler, all these kinds of things. Words we use in English all the time are rather colorful words in England. Mm-hmm. I won't give you any specific examples, right. But it's really funny.
Well, that is awesome, and great to hear that you've had so many opportunities to travel the world, to go to different places. And I think getting exposed to these different cultures is a really great aspect of careers in science, as you do get these opportunities. You get to work with a variety of different people, and it tends to be a relatively friendly environment for this learning process to happen in.
And also, when you're visiting someone scientifically, they take care of you. The times I've been to China, I was well cared for. People show you around. You feel as if you instantly have a circle of friends. And you see things that you wouldn't normally see. You go places. So if you go as a scientist, you see things you wouldn't normally see. I spent 2 days at the Palace Museum in Taipei, which was amazing. They only show something like 3% of their collection. And I barely got through that in 2 days.
Wow.
So you have an opportunity as a scientist to see things you wouldn't ordinarily see as a regular tourist. So it's really great.
Definitely. And I think we've hinted at this last part of our conversation about some of the people that you get to work with in science, and they're phenomenal. I think there are so many creative, funny, brilliant people that you get to work with. And in these different lab groups, there are a lot of different personalities that spring up, and sometimes it brings along with it quirky traditions or just hilarious memories that you've shared together. Because when it comes down to it, science is done by people, and you like to have fun with your work. So can you share with us today, Jan, one of your own maybe funny science memories or a quirky tradition that you've experienced?
I'm really big into practical jokes.
Awesome.
I remember once when I was working at Eli Lilly, I managed to get the stationery from the radiation safety office, and I wrote my boss a letter on stationery from the radiation safety office about having his coffee cup on the lab bench. He literally freaked out.
Oh, no.
Of course, all a joke.
Right.
And he actually called the radiation safety office to try to explain about his coffee cup, and they didn't know what he was talking about.
Oh, jeez. That went pretty far. I'm impressed.
When I was at Harvard, we managed to get stationery from MIT and write letters to faculty members. I mean, it was nothing dangerous or bad. I think one of the worst things I ever did, I took a piece of foam, like the packing foam that you have in boxes, mm-hmm. and I iced it with real icing for Bill Resnikoff's birthday. And he's such a nice guy. And I preambled this by saying, I'm a terrible cook. This is my first cake I've ever made.
Oh, wow.
So he spent 10 minutes trying to cut into this thing. And it was real icing on the outside mm-hmm. before he gave up because he didn't want to embarrass me. And it was hilarious. You know, he got me back.
Right.
In my own lab, we have holiday parties around New Year's. And the rule is you have to give someone a present that's really embarrassing, and they have to display it on their desk for a year.
Wow, this is high stakes.
Trust me, it's high stakes. And I've been the recipient of some things that I had to put behind the door of my office.
Right. Oh, wow. So is there anyone that you could share on the show?
Well, I have a picture of me and George Bush, autographed, photoshopped in together, obviously. And it was, if you knew me, you would know that hanging that in my office was difficult.
Gotcha. Well, I love this whole picture that you're painting of your lab group just being kind of a fun, sort of practical, jokey. You take your work seriously, but you're able to enjoy each other's company as well. And I think that's so important for a happy lab.
It is.
Well, thank you for sharing your stories with us today, Jan. And I know we've talked about your research throughout the interview, and I want to ask you a tough question next. This is one of my favorite questions to ask people, basically about the big questions that are left unanswered for them. So, Jan, if I gave you everything today, if things like funding, staff, technology, feasibility, not a problem, what is the question that you would want to answer?
It's exactly what I'm doing. And the big challenge in genetics for me is taming microorganisms, taking these organisms from the environment that do things that we will never be able to recapitulate biochemically, and teaching them to do the things we want them to do. When I was a graduate student, I went to a seminar by William Hayes, who wrote the Bacterial Bible, and he said something that really impressed me. He said, if you're smart enough to ask an organism a question in a way that will allow you to understand the answer, it will always give you an answer, and it will never lie to you. And I think of genetics as a language that you develop with the organisms you study. And there are so many incredible organisms out there that have so much to teach us. E.O. Wilson said that if he were starting his career over, he'd become a microbiologist so he could have that dialogue. And I think that's just fascinating to be able to study these organisms that are so untapped. There's so much we don't know.
Definitely. And I really like the way you've put that. I've never really thought of science of genetics as a language, as a way of really getting answers from and communicating with the natural world. So very, very cool. Now, we also like to acquire advice from the different people we've had on our program. So I'm going to trouble you today, Jan, for a piece of advice. Can you think back on all the advice you've received over your career and share with us what you think is the best gem?
There are two related pieces of advice that I was given. When I was a beginning assistant professor, Rich Losick told me to choose the people in my lab carefully. He said, you're better off sitting at a bench by yourself than surrounding yourself with people who are not going to be productive, are not going to make the kind of investment you want from them. And I followed that advice. I don't run a big lab. I've trained graduate students, but my lab is like the Marines. It's a few great people.
I like it.
We're a team. We have our oars in the water, pointed in the same direction. And choosing the people in your lab, you're not just creating an environment for them, you're creating an environment for yourself. And being happy is important in that environment. Related to that, Greg Petsko told me years ago that you have to choose your collaborators with as much care as you choose your friends. And that has been an invaluable piece of advice as my career has progressed. And it's become more and more important to me as my career has gone on. Collaboration in science is all about trust. And choosing people that you can trust and work with, who have their eye on the science, not on their own egos and not on their own self-aggrandizement, but people who care about science as much as you do. And that piece of advice has really stood me well.
Well, these are both fantastic pieces of advice, Jan. I think the company you keep in science is important. So building the lab with the people that you want to be there who are also equally invested, I think is great advice. And then thinking about that from a collaborative point of view as well, very important. So I think you've shared some wonderful pearls with us today. Jan, can you share one last piece of advice or maybe one last note of inspiration that you would like to end our interview on today?
This is advice I give to my students and to the undergraduates I teach. Do what you love. Don't do what's practical. Don't do what your parents think you're going to get a job doing. Do what you love to do, because you're never going to be happy and you're not going to be successful if you don't follow what your gut and your heart tells you you care about. You're not going to invest in learning about something unless you really care about it. You're not going to make the kind of sacrifices it takes to be really successful at something if you don't honestly care about the outcome.
Well, very well said, Jan. I like it. Wonderful note to end on. Now, can you tell our listeners how they can reach out if they want to learn more about you and what you're doing?
Well, I am one of the few Luddites left on the planet. I don't tweet. I don't blog. I'm not antisocial about social media. But the university has a website, and I have an email address, and I'm always happy to correspond by email. I have a small web page, but I think these things can just consume your life if you allow them to.
This is true.
So my phone has an off button, which I tell my husband on a regular basis. You can turn the thing off.
I think mine's gathering dust as we speak. But Well, Jan, wonderful to hear how we can get in touch with you. Listeners, definitely check out the website. Send an email if you have any questions for Jan. And Jan, thank you so much for joining us today and sharing your story.
Thank you. This has really been a lot of fun.
Well, it was a pleasure for me to chat with you today. I've really enjoyed our conversation. And listeners, we hope you've enjoyed our conversation as well. And we'll see you again next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.